Alright guys, just a couple things before we get started here today. First of all, this is the answer key to your homework from chapter 16 and 17 comparing DNA replication, transcription, and translation. So make sure you look over this and ask me any questions you have. Second of all, the video today is basically what I would write on the whiteboard as we were going over the slides. So make sure you have the lecture slides open to accompany what we're going through today. We're going to start on chapter 18 on regulation of gene expression. We're first going to talk about prokaryotic gene regulation. That's in bacteria, not in us, not in eukaryotes. The regulation that we talk about here doesn't really happen in eukaryotes, with a few small exceptions. In prokaryotes, genes that work together on a related function are grouped together in a cluster called an operon. The operon includes both the genes that are regulated and the promoter and regulatory sequences in front of the cluster of genes. Within the promoter, there is a special stretch of DNA that's a binding site for a protein called the repressor, which blocks gene expression, and that's called the operator. The operator is the DNA that's a binding site for a protein that blocks gene expression. The protein that blocks gene expression is called a repressor. Some repressors need a cofactor to help them function, and so in order to turn the repressor on, some repressors require a co-repressor. The first type of operon we're going to talk about as an example is the trip operon. It's the cluster of genes that are responsible for synthesizing the amino acid tryptophan. This is a repressible operon. What that means is it's normally on because bacteria need the amino acid tryptophan, but it can be shut off. If there's an excess of tryptophan, then it would be wasteful to make the enzymes to make more of something there's already an abundance of. For the trip operon, if there's no tryptophan, an amino acid which the bacteria need, then the default setting is the operon is on, and so it starts making the enzymes that generate more tryptophan. Tryptophan itself is the co-repressor, so as tryptophan builds up, the co-repressor builds up. And if there's an abundance of tryptophan, the co-repressor, it binds to the repressor. When the co-repressor binds the repressor, it activates the repressor, turning it on. And when the repressor is active, it binds to the operator. Now you have a protein bound to the promoter which blocks RNA polymerase from being able to transcribe those genes. And that operon is shut off. It's repressed. Now we're going to switch and talk about a different operon. This is the lac operon. It makes the enzymes that are used to break down the disaccharide lactose. This is an example of an inducible operon. It's normally off, but it can be turned on. If there's no lactose sugar around, it would be wasteful to make the enzymes to break it down. But when lactose becomes present, this operon can be turned on. The way that the lac operon is turned on is by the presence of an inducer. The inducer is a small molecule that blocks the function of the repressor protein. And in the case of the lac operon, the inducer is allolactose, which is just an isoform of lactose itself. If there's no lactose, there's no isoform of lactose, there's no allolactose. If there's no lactose, there's no inducer. If there's no inducer, the repressor protein in this case works by itself. It doesn't require a cofactor to function. And if the repressor protein is active, it binds to the operator, blocking transcription, and the operon is turned off. If lactose is present, then the inducer is present. If the inducer is present, it inactivates the repressor. It stops the repressor from functioning. So in the presence of the inducer, the repressor does not bind to the operator. And without anything bound to the operator, the operon is on and the genes are expressed. Both the trip operon and the lac operon are examples of negative gene regulation because they use a repressor. But for the trip operon, it's a repressible operon. It's normally on, but it can be shut off by the presence of the co-repressor. Repressible operons are often involved in anabolic pathways that are used to build something up, and then when there's an abundance of it, that end product is the co-repressor that shuts the system down. The lac operon is an example of an inducible operon. It's normally off, but it can be turned on. And inducible operons are typical of catabolic pathways, where they're only turned on when the thing that's going to be broken down is present. The lac operon also has positive gene regulation. It has an activator protein called CAP, or catabolite activator protein, that increases transcription to a high, robust level. And CAP requires a cofactor called cyclic AMP. Cyclic AMP is the breakdown product of AMP, adenosine monophosphate, which is the breakdown product of ADP, which is the breakdown product of ATP. So what it means for the cell is if there's an abundance of cyclic AMP, all of the energy has been used up. The lac operon can be both positively and negatively regulated. And what that's doing is allowing the bacteria to choose between different food sources. So glucose is the bacteria's favorite food because it can go straight into cellular respiration. Lactose is their second choice because there are several steps that have to happen before the lactose can be metabolized in cellular respiration. So we're going to go through the different combinations of what happens in the presence of glucose or lactose or both or neither. First is the case where glucose is present but lactose is absent. So again, glucose is the bacteria's preferred food. If there is glucose, then cellular respiration is occurring and all of the AMP is turned into ADP, is turned into ATP. And so all the cyclic AMP is used up to make lots of ATP. Without any cyclic AMP, CAP is not activated. In this case, lactose is absent. If lactose is absent, then the inducer is absent. If the inducer is absent, the repressor is active. If the repressor is active, the repressor can bind to the operator, and the operon is off. Now let's think about what happens when both glucose and lactose are available to the bacteria. Again, if glucose is present, then all the cyclic AMP is used up, and if there's no cyclic AMP, CAP is not active. And so we don't get any activation of the lac operon. If lactose is present, that means that the inducer is present. If the inducer is present, the inducer blocks the activity of the repressor, so the repressor is off. If the repressor is off, the operon is on. It's on, however, it's only at kind of a low basal level. It's not being expressed robustly. If glucose is absent and lactose is present, however, something slightly different happens. If glucose is absent, then the ATP stores start dropping and cyclic AMP increases. If cyclic AMP is present, then that means that CAP is active and can activate transcription. At the same time, because lactose is present, the inducer is present, and the inducer is blocking the activity of the repressor. If the repressor is inactivated, then the operon is on. Now we have the operon able to be expressed because of the presence of the inducer, and now being robustly expressed at a high level with the help of CAP, our activator protein. But what happens if both glucose and lactose are absent? If there's no glucose, then there is cyclic AMP, and our CAP activator protein is active. However, that doesn't really matter if there's no lactose. If there's no lactose, there's no inducer. If there's no inducer, then the repressor is active. And if the repressor is active, then the operon is off. If there's a competition between an activator protein and a repressor protein, the repressor wins and blocks transcription. One thing to mention about this chart, it's not my ideal for you guys just to memorize this chart. Instead, you need to understand the relationships between inducers and repressors and transcription of the operon, and also the co-repressor which functions in the trip operon. So understand that inducers turn the operon on. They induce it by turning the repressor off. And so we have all these double negatives. And I want you to be able to work through and figure out, if I tell you the repressor's active, what does that mean? Is the inducer present or absent? If the repressor's active, is the operon on or off? And so you should be able to work through and understand the relationship between these proteins and gene expression.